Hello everyone, I'm back with another walkthrough. We will go through Biology Paper 2, Higher Tier or AQA Combined Science. This is the 2019 paper and your exam takes place tomorrow in the afternoon. Okay, so question 1. Some students investigated the effect of drinking caffeine on reaction time. They used a drink containing 32.25 milligrams of caffeine per 100 centimeter cubed. This is the method used. Divide the students into 4 groups, A, B, C and D. Measure and record the reaction time of each student using the ruler drop test. Students in Group A drink 200 centimeter cubed of water. Group B drinks 200 centimeter of the caffeine drink. C drinks 400, D drinks 600. So as we move from B to D, we are adding 200 centimeter cubed of the caffeine drink. Repeat steps 2 after 15 minutes. Describe how to do the ruler drop test. So this is your required practical. So it's 3 marks. So here we are not looking for a lengthy method. So keep it nice and straightforward. So first bullet point, and I keep saying this in my walkthrough videos, it's okay to write in bullet points. Hold the ruler. So hold the ruler above the hand of the other student. Drop the ruler. Obviously, you won't be telling them that you are dropping the ruler. So drop the ruler and the other student will catch it. And your final bullet point is record where the ruler is caught. So remember, if you think back to when you did this in your lessons, the measurement is taken above your thumb or your index finger. That should get you 3 marks. Question 1.2 shows the mass of caffeine taken in by each student. So remember, Group A was the one that had 200 centimeter cubed of water. This one, Group B, got 200 of the caffeinated drink. This one was 400 caffeinated drink. And this one took 600. So how can you work out the mass of caffeine? Well, 200 plus 400 gives you 600 centimeter cubed. So what you need to do is, to get the mass, is you just add these 2 values up. So 64.5 add 129 gives you 193.5. Why did Group A drink water instead of the caffeine drink? So Group A is basically acting as your control. This allows you to then compare your results with B, C and D. So how would you structure your answer? You can say Group A drink water because it acts as a control, or you can say to compare the effect of no caffeine. So to compare the effect of no caffeine. So this allows you to compare the reaction time for someone who drinks 200 centimeter cubed of water with 3 other groups who have drank a caffeinated drink. Table 2 was used to convert the results of the ruler drop test into reaction time. Okay, you're not expected to memorize these values for your required practical. Your conversion tables will be given. Estimate the reaction time for a student who recorded a distance of 23 centimeters. So we're looking at, somewhere here, so 23. So what you do here is work out the difference between these two values, yeah? So that's the highest reading. So you go 0.212 minus 0.202. So I'm working out the difference. That is 0.01. So basically I'm trying to show you how you're working out the difference between 21 and 22. Difference between 20 and 21. Once I find that increment, I can then add it on here. So the difference is 0.01. I'm going to divide that by 2 because it's halfway, it's a middle reading. Yeah. And then I'm going to add that value, which is 0.05, onto this value. And then that gives me 0.217. So I'm just estimating. So that 23 centimeter, my reaction time is 0.217. Students calculated the decrease in their reaction time after the drink compared with before the drink. Figure 1 shows the results for each student. Describe the effect of the mass of caffeine taken in on the decrease in reaction time. So on your y-axis, you've got decrease in reaction time in seconds. And I know group A has 0 caffeine. That one had, so that one was 64.5. I'm just going back and taking the readings from that table. That was 129. And this one we calculated 193.5. So what's happening to the decrease in reaction time as I increase the mass of caffeine? And we can see that the values are obviously going up. So how would you phrase that? You'd say as the mass of caffeine increases, and then just take your label on the y-axis, the decrease in reaction time in seconds increases. It's 1 mark. For 3 students, the decrease in reaction time was negative. So negative, negative, negative. Give the reason why the value was negative. Because their reaction time was greater, which is why the decrease then becomes negative when you work out that difference. What is the range of the results for group C? So your range from maths, you should remember this, is the difference between your highest value and your lowest value. So if we go to group C and read these values off, so my highest is, this is my highest point, and there's my lowest point. Let's start with the lowest point. So if you read that across, it's roughly halfway here. So that's 0.01. So my lowest reading is 0.01. go back up, read that value off, that's my highest point. So my range is 0.01 to 0.18. You can leave it like that, or you can work out the difference. So put that in your calculator and you can work out the difference, or you can just leave it like that. Suggest two variables that should have been controlled in this investigation. So we're testing how caffeine affects reaction time in students. So what kind of variables should we keep the same? So we need to make sure that all the students are the same age, okay, and they must be the same gender. It's nice and straightforward. I don't want you to think, oh, I've got 4 lines here, I need to write a lot. You don't need to. Other things that you could have is you can have height, weight, any medications that they took earlier on during the day or before the experiment. They need to be equally tired, or all of them should have received the same amount of sleep. You can't have someone here with 2 hours of sleep and then you've got someone else who had 10 hours of sleep, because that would have an impact on your reaction time. Practice of the ruler drop test. If someone's practiced and practiced, then obviously they would be better. Their reaction time would be lower. What else could you have? You can have using the same hand. So if they've used the right hand, it has to be the right hand for everyone. Number of students in each group, and starting point of ruler. Obviously we're assuming that 0 is towards the hand. 1.9. Explain why the ruler drop test does not involve a reflex action. So does not involve a reflex action. So you say it's not automatic, and remember, a reflex action involves the spinal cord, because a reflex action tries to protect you from harm. But here it's saying, oh, it's not a reflex action. That means it does not does involve the brain. It's not a reflex action, involves the brain. When the brain is involved, the reaction time should be longer, and when the brain is involved, we say it is not a reflex action. So here, this question is basically testing you on, do you know your definition of reflex action? So reflex action is an automatic response, and it does not involve the brain. So just double check it, read the question again. Even I was writing does not. The not is in bold. So it does not involve the brain. That's basically what reflex action is. But here we need to write why the ruler drop test is not a reflex action. So we write the opposite. There has been a rapid increase in the percentage of CO2, carbon dioxide, in the atmosphere since 1960. Carbon dioxide is a greenhouse gas. Name one other greenhouse gas. Methane. Okay, and then the third one is water vapor. We just need to write one. Global warming causes climate change. Give 2 effects of climate change. So we can say sea levels rising, ice caps melting. Keep it nice and simple. No need to waffle, no need to give a lengthy essay. Not interested. 2.3. Plants take in carbon dioxide from the atmosphere. Figure 2 shows part of the carbon cycle. So this question, if you have a look, they've given you a whole page to write the answer. So you might think, oh gosh, you know, what am I writing here? But you're very lucky, there is a diagram here. So use the diagram as your plan. So let's have a look at what all of these arrows mean. So carbon cycle shows you how carbon dioxide is recycled through the atmosphere and then back on land and so on. So carbon dioxide, this arrow here from the animals is going into the atmosphere. So that's saying that CO2 is being produced by animals. What's the name of that reaction? It's respiration. Animals to microorganisms, that must be animals dying and decaying. Then I've got microorganisms, an arrow going to the atmosphere, so that must be microorganisms such as bacteria also respiring. Green plants into animals, that means animals are feeding. CO2 going into green plants, that must be photosynthesis. And then carbon dioxide in the atmosphere is taken in, so you can see an arrowhead there and an arrowhead there, so that must be, sorry, I didn't see the green plants there, but green plants into microorganism, that must be decay, and then green plants going into the atmosphere, that is respiration. Remember, plants also respire. So that's your planning done, and all you need to do now is add that as bullet points and use each of these as your headings. Describe how carbon from the atmosphere is cycled through living organisms. So here, let's start off with photosynthesis. That's my first heading. And I have said this in my previous videos, it's for a 6 marker. You are allowed to have subheadings and bullet points. So I can say here, photosynthesis, carbon dioxide is entering leaves via stomata, and it's being used for photosynthesis to make glucose. That should be enough for photosynthesis. Let's move on to tick off each process once you've covered it. Let's go to feeding. So for feeding, you can say plants are eaten by animals which, so what animals are doing with that plant material, they use carbon compounds to make other carbon compounds. Let's talk about decay. Animals die. They are decayed by microorganisms. That should be enough. And then respiration. So here, plants, animals, and microorganisms respire to release CO2 back into the atmosphere. So here all I've done is I've used my diagram, and this is really, really helpful. So to recap, a question like this, if you're given the carbon cycle, label each arrow and then you just write about each process that you've added on to your diagram. So not a difficult question, it might come across as difficult, but it isn't. 3.1, vectors are used in the process of genetic engineering. Which two statements are correct? So vectors are enzymes. No, they're not. Vectors are used to insert genes into cells. Yes. Vectors are used to isolate the required gene. No, because that's enzymes do that. Vectors are used to stimulate cell division. Nope. Vectors are usually plasmids or viruses. Yes. So now plasmid is your circular strand of DNA, which is obtained from prokaryotic cells or bacterial cells. Scientists have genetically engineered a variety of wheat to be resistant to herbicides. So herbicides are chemicals that kill weeds. So this variety of wheat is resistant to herbicides, which means that if you spray herbicides, it won't damage your wheat plant. A wheat plant is used to make flour, which is then used to, say, make Weetabix, for example. The herbicide-resistant variety of wheat will give a higher yield, so yield means the amount of product, than non-herbicide-resistant variety. Non-herbicide-resistant variety would mean if I spray the herbicide, it would kill the wheat. Explain why. So here you would say wheat is not affected by spraying herbicide. So wheat gets more light. So there is more photosynthesis. It's an explain question, so you need to explain using science, which is this bit here. Wheat gets more light, more photosynthesis, or so there's more photosynthesis, or it can or the plant produces more glucose, you can even say that. Give two examples of genetic engineering in use today. Do not refer to the example that's already been given, which is herbicide resistance. So here we remember one of the things on the spec is production of human insulin, and then you can say frost resistant crops. So normally plants get damaged when it's really frosty. So if you've got frost resistant crops, they can grow and not be impacted by the frost. Other answers you can have: crops resistant to diseases, crops resistant to pests, crops resistant to drought, crops with added nutrients. So for example, in your lessons you should have studied golden rice, which has the vitamin A gene. So when people consume the golden rice, their bodies are able to make vitamin A. Plants or crops with more bigger fruits or higher yield, crops with improved taste, or crops with improved shelf life. Okay, so there's quite a lot on the mark scheme that would allow you to pick up marks. Scientists working on the Human Genome Project have now mapped the entire genetic code of humans. Explain one way this could be improved for people in the future. Definition of genome is your entire genetic material. Basically all your 46 chromosomes. How do I get hold of that? I just need access to one body cell, and I can extract all of this from the nucleus of this body cell. So for example, if I take a saliva swab, I've got access to your cheek cells, and if I just take one cheek cell and get hold of the nucleus, I can extract that genetic material. So why is this important? Apologies, I was looking for the spec because I want to show you something. So if you look at the AQA spec, it clearly states the purpose of the Genome Project. So, where are we? So if you look at the spec under DNA and the genome, it says students should be able to discuss the importance of understanding the human genome. This question is related to this. This is limited to which means that students need to know these 3 bullet points. Search for genes linked to different types of diseases, understanding and treatment of inherited disorders, use in tracing human migration patterns from the past. And it's this that's on the mark scheme, always. You don't need to write anything else, you just write about, basically, remember these bullet points and you write the same thing down. If you want access to the spec, it's freely available on the AQA website. Make sure you select Combined Science and its Trilogy. So, the first bullet point, I'm going to say what was on the spec. Tracing human migration patterns from the past. And second bullet point, it's 2 marks, identifying genes that cause inherited disorders. And leave it there. Question 4.1 looks at the topic adaptations. You need to know, so if you look at the spec, again, it doesn't say you need to study, say, the camel in detail or the polar bear in detail. It's quite generic. So if I show you, so this comes under ecology. There we go. So students should be able to explain how organisms are adapted to live in their natural environment when given appropriate information, which is what we've got here. Organisms have features, adaptations, that enable them to survive in the conditions in which they normally live. These adaptations may be structural, so structural adaptations, behavioral, or functional. Functional is linked to what's happening in the actual cell. And then it goes on to extremophiles. So they've given us the information. We've got, it's supposed to be a bird, a male palm cockatoos use sticks to beat. So they use sticks to beat. That's them behaving in a particular way. So that's a behavioral adaptation. The harmless hornet moth has black and yellow stripes to look like a bee or wasp. That's to do with their structure. Sea spiders have automatic muscle contractions that move oxygen around their bodies. This is functional because it's happening inside the body. So that's to do with how they behave, how they look, and inside the body. Things that are happening that you can't see. Plants also have adaptations. There are more than 28,000 known species of orchid plants. Many plants grow attached to other types of plants, brightly coloured flowers, large quantities of pollen, thousands of tiny light seeds are produced. Now what you need to do here is describe how each of these adaptations help the orchid plant to survive and compete. It's 4 marks. So you just need to write down 1 bullet point for each. So I would go 1, 2, 3, 4, and I would go number 1. So for number 1, I would say, right, grow attached to other types of plants. Well, that's to do with support. So support provided, why? To absorb more light. For what? For photosynthesis. And just go back to the question, it says to survive. How these adaptations help plants to survive and compete. 2. Brightly coloured flowers attract pollinators, such as bees. But you can just leave it like that. Number 3. Large quantities of pollen produced, that increases the reproduction. More reproduction will occur. Produce thousands of tiny light seeds. Light seeds can travel long distances to grow in new areas. This is survival. Question 4.3. A rare orchid has been found in the mountains in China. Pale yellow flowers. DNA analysis of the genome shows that it is an ancestral species. So it existed thousands and thousands of years ago. All other present-day orchids evolved from this ancestral species millions of years ago. So not thousands, millions of years ago. One present-day species has bright purple flowers. So this is at present. Describe how an orchid with bright purple flowers may have evolved from ancestral species which has pale yellow flowers. So what's the underlying topic here? It's evolution by natural selection. So here it's all about surviving, breeding, passing on that beneficial gene. So for 4 marks you would start off by saying how purple flowers came about. So that could be genetic variation down to mutation. So you can just say genetic variation gives rise to purple flowers. Then what? These survive and breed. If they breed, what does that mean? They will pass on the purple color gene. Don't just say, oh, they pass on this beneficial gene, because in this question they've told you that it's purple colored flowers. Then what happens is they become so different that they can no longer interbreed with ancestral species. Okay. 4.4. So the DNA code codes for proteins, which means the DNA code must be coding for amino acids, which then codes for protein, and it's this protein that gives rise to flower colour. So I've got ancestral species, my 4 species here, and I've got the amino acid sequence. Okay, this is just a shorthand way of representing amino acids. Suggest why orchid species is most closely related to the ancestral species. Give a reason for your answer. Which is most closely related to this species? So, close relation would have almost identical DNA sequence, which leads to an identical amino acid sequence. So let's have a look at these species and try to identify if there is a difference in the sequence. So let's have a look at Allah. Right, so that's different. Okay, so this is very different. Have a look at this one. Allah, Lu, Allah, Isolu, Targli, Allah. So we've got that as a difference, that has a difference. Okay, so there's 2 differences there. And there's quite a few here. Allah, that's different. So I'm comparing it to this one. That's there, that's different, that's different, different. Okay, there's quite a few here. So look at that. Allah, yep, that's there, that's there, that's there, that's there. Allah, Lu, Bly, Allah. So if I compare this one with that amino acid sequence, there's only one different amino acid, the TYR. So because the amino acid sequence is almost the same, there's only one amino acid that's different, we say D is closely related, and your explanation is basically what you've detected. Only one amino acid is different. So this obviously is not on the spec. This is done in depth for triple science, but the reason why it's on combined science is because this is an application question. The information has been given to you. Okay, you just need to spend some time reading, okay, and checking for those differences before you get to your answer. Right, question 5. Some students estimated the population of daisies in a school field. So this is your ecology required practical, which looks at sampling using quadrats. Find a place where daisies are growing, put the quadrat down. Now here it doesn't really tell you how to put the quadrat down, but you should remember from your studies that you use a technique which allows you to place the quadrats randomly. Count and record, repeat steps, calculate the mean, use the data to estimate the total number of daisies. So see there's calculations involved here. Which 2 improvements would increase the validity of this method? So do not put any quadrats in the trees. No, repeat for another 10 quadrats. The more repetitions, the better. That doesn't help. Random method. Same person to place all the quadrats. Nah. So it's these 2 that are the best answers. Okay? Right, next the question is 5 marker, which is a calculation question allowing you to estimate the population. So calculate the population of daisy plants on the school field. Right. So we are placing a quadrat. So remember you get different sized quadrats. You can have 1 meter by 1 meter, which gives you an area of 1 meter squared. This quadrat here, first of all, convert that to meters, so that's 0.5 meter by 0.5 meter, gives you an area of 0.25 meter squared. So that's a smaller quadrat, gives you a smaller area. Basically, you would need all of these to fit in to one of those. And in one quadrat, so in this 0.5 meter by 0.5 meter, I end up with 7.65 daisy plants. Right. So, how do I calculate the population of daisy plants on the school field? Your first step is work out the area of the school field. Okay, so that's what we're going to do, area. Look at your diagram and you can see it's a strange looking field. It's not a square, it's not a rectangle, but if you have a look, it looks like I've got a rectangle, so also use a ruler here, and then I've got a triangle. So the length here is 62 meters, that's 164 meters, so the area for the rectangle bit, which is its length times width, so 62 times 164 gives me that meter squared. Then I've got my triangle, so the base is 164, and I need to work out the height here. That's not that complicated, so the entire length, so the entire height here is 130. If I subtract 62 from that, that should give me, get hold of a calculator, 68. 68 times 164 times 2, so my area of triangle is base times height divided by 2. Put that in your calculator, you should get 5,576 meters squared. So my total area is, add those two values up, and that gives you an area of 15,744 meters squared. Right, so I've done the area. Right. So I've got a quadrat, that's 0.5 by 0.5, which gives me 0.25 meters squared. And in one quadrat, I have 7.65 daisy plants. And what I need to do now is find out how many of these can I fit onto the school field. There's my total area. That's the area of one quadrat. So I should divide. So 15744, divide that by 0.25 meters squared, meter squared. That should give me the number of quadrats that could fit on that school field. So that is 62976 quadrats. And in the question it said, in one quadrat, it was the mean number of daisy plants per quadrat was 7.65. So all I do is I multiply the number of quadrats by the number of daisy plants. That gives me 481,766.4. You can't have 0.4 over daisy plant. Now what you need to do is go back to the question again. It says give your answer in standard form to two significant figures. So you get a mark for standard form, you get a mark for two significant figures. Right, first of all let's work out significant figures. That's 480000. Okay, we'll get rid of that. Let's have a whole number because you can't really have a 0.4 of a daisy plot. Right now, standard form, but to two significant figures, so 4.8. So if my decimal place is there, how many places would it have to move to get to the end? 1, 2, 3, 4, 5 times 10 to the power of 5. And that's your answer. .3, the students noticed a very uneven distribution of daisy plants in the field. Explain how different biotic factors and abiotic factors could have caused an uneven distribution of daisy plants. Use figure 3 on page 20 to help you. So, you've got a school building there, you've got trees there, there's a gate there. So biotic factors are living factors, and abiotic are your non-living things such as water, soil, rocks, oxygen, carbon dioxide, living things, predators, prey, animals, plants, pathogens, diseases caused by pathogens. So explain how different factors could have caused uneven distribution of daisy plants. Look at the diagram, and as I said, there's trees. So what? How would that affect distribution? Well, that reduces light for photosynthesis. That means fewer daisies in this area. Or you can say trees take more water, so that means there's not enough water for the daisy plants, so fewer daisy plants. So this is looking at competition. And then the school is right next to obviously the field, so you can say trampling will kill the daisy plants. So fewer daisies here. What else can we have? We can have the school building. So school building is huge. What could happen with that? So reduces, so because of the shade, there's a reduction in light, reduces light levels. Less photosynthesis, fewer daisies here. You get the idea. Let's do one more. So in some areas you might get more animals. I don't know, like squirrels maybe. What would those do? They might eat the daisies. What's the impact? Fewer daisies. Okay, so if you get a question like this where you are discussing biotic and abiotic factors and it's an explain question, this is how you would structure your answer. Question 6 is now moving on to look at homeostasis. Define the term homeostasis. So it's all about maintaining a constant internal environment, so control of internal conditions. Y for optimum enzyme activity. Name the hormone released if the blood glucose concentration falls too low. Possibly because you've been fasting, say for 10 to 12 hours. It is glucagon, not insulin. Insulin is released if blood glucose concentration is too high, which is after a meal. Make sure you get the spelling right. 2 people were sent to a hospital to find out if they have diabetes. This is the method used at the hospital. Do not eat or drink after midnight. This is fasting. Measure blood glucose at 9:30. Drink a glucose solution at 10. Measure blood glucose concentration for the next 3 hours. Which is what we're doing here. Okay? So person A and person B have diabetes. Describe how figure 4 shows that person B has diabetes. Use data from figure 4. So look at the results and you can see that it says do not eat or drink after midnight. This is called fasting, right? So look at this bit here. This is the glucose concentration after fasting, okay, from midnight onwards. So you can see that the fasting blood glucose is higher than the normal range. Well, how do I know what the normal range is? Look at the graph, it's been given to you here. It shouldn't be more than 5.5, but it's above 8. Okay? Any information that you are given in an exam question, there is a purpose behind it, so you must use it. And then if you look, this is where they have that glucose drink. Yeah? And you can see how the glucose concentration falls slowly back to 12. So glucose concentration fell slowly after reaching peak. That's 2 marks. Now where's the third mark? Quote a value from the graph. So I'm going to do that now. Peak 12 millimoles per dm cubed. So 1 mark here, 1 mark there, and that's my third mark. Person A and person B had a test to measure the concentration of insulin in their blood when they were fasting. Table 4 shows the results. Fasting blood insulin concentration. So this person's fasting, but look at how high the insulin levels are. Person B, that's low, and that's your normal range. Okay, so this one's below, that one's way above 175. Suggest which type of diabetes person A and person B have. Give a reason for each answer. Now person A, they have plenty of insulin, so this probably means that the body cells of person A are not utilizing that insulin, but they are producing it. So that type of diabetes is type 2. Remember, type 2 diabetes shows up in late adulthood, so around about the age of 40. Now, what's my reason? Insulin levels are high, but the issue is the body cells are not responding. Person B, insulin levels are low, which probably suggests that this person isn't making insulin. Issue with the pancreas. So type 1, not producing enough insulin. And then our last question is an application question. It does come across quite challenging. So toxic hypoglycemia syndrome has caused the deaths of hundreds of starving children. The starving children have had nothing to eat all day. The starving children then eat many lychee fruits. The lychee fruit contain a molecule which stops an enzyme in the liver working. This enzyme normally converts stored fats into glucose. Children who have eaten during the day are not affected by eating the fruit. Starving children die when they eat the lychee fruit. But children who have eaten, starving children may die from eating many lychee fruits, but children who have eaten during the day are not affected. Okay? Let me just go back. Children who have eaten during the day are not affected by eating many lychee. Okay, so basically the question is saying that children who eat lychee fruits die, and the ones who don't eat survive. They're not affected. So your first bullet point is looking at existing glycogen stores. So starving children have no glycogen stores because they've already used that up, yeah. So they need glucose from fats, yeah. But if they have the lychee fruit, there's an enzyme there that stops the liver from working. So when they eat lychee fruits, there is a molecule which stops the enzyme that converts fat into glucose to stop working. So this enzyme here that does this no longer works in children who have eaten the lychee fruit. So enzyme is stopped from working, so they have no glucose from fats. So there's no glucose. How would that lead to death? No glucose means no respiration. So obviously no respiration, no energy is released, so they will die. So no respiration leads to death. So I've explained why children who eat the lychee fruit die, but what about the others who have lychee fruit and who are eating other food? So you would say children that, the children who have eaten, so children that are not starving and have eaten have glycogen store in the liver or muscles, and glucagon continues, which is your hormone, convert glycogen into glucose. Okay, so this, when you read it, you're probably thinking, gosh, this is very challenging, or my teacher hasn't taught me this. Yes, they have. This is your last topic of biology paper 1, bioenergetics, looking at respiration and not biology paper 1, this is biology paper 2, which looks at your it's the homeostasis topic, looking at blood glucose concentration. Okay, and it's looking at, can you apply your existing knowledge? So if you get a question like this and you don't know how to answer it, the best way forward is think, what's the underlying topic in a biology paper 2 that would help you to answer this question? Okay, hope you found the walkthrough useful, and I hope your exam goes well tomorrow, and I look forward to talking to you all again in the next few days, because I'm planning on putting up the chemistry walkthroughs for chemistry paper 2 and physics paper 2. So I shall leave you to it. Hope you find this video useful. Speak to you soon.